Horizontal Gene Transfer, The Hidden Hazards of Genetic Engineering by Dr. Mae-Wan Ho. Genetic engineering involves designing artificial constructs to cross species barriers and to invade genomes. In other words, it enhances horizontal gene transfer, the direct transfer of genetic material to unrelated species. The artificial constructs of transgenic DNA typically contain genetic material from bacteria, viruses, and other genetic parasites that cause disease, as well as antibiotic resistance, genes that make infectious disease untreatable. Horizontal transfer of transgenic DNA has the potential, among other things, to create new viruses and bacteria that cause diseases and spread drug and antibiotic resistant genes among pathogens. There is an urgent need to establish effective regulatory oversight to prevent the escape and release of these dangerous constructs into the environment, and to consider whether some of the most dangerous experiments should be allowed to continue at all. Transgenic pollen and baby bees. Professor Hans Heinrich Katz from the University of Jena is reported to have new evidence, as yet unpublished, that gene engineered into transgenic plants have transferred via pollen to bacteria and yeasts living in the gut of bee larvae. If Professor Katz's claim can be substantiated, it indicates that new genes and gene constructs introduced into transgenic crops and other transgenic organisms can spread, not just by ordinary cross-pollination or crossbreeding to closely related species, but by genes and gene constructs invading the genomes, the totality of the organism's own genetic material, of completely unrelated species, including the microorganisms living in the gut of animals eating transgenic material. The finding is not unexpected. Some scientists have been drawing attention to this possibility recently, but the warnings actually date back to the mid-1970s when genetic engineering began. Hundreds of scientists around the world are now demanding a moratorium on all environmental releases of transgenic organisms on grounds of safety, and horizontal gene transfer is one of the major considerations. Some of us have argued that the hazards of horizontal gene transfer to unrelated species are inherent to genetic engineering. The genes of gene constructs created in genetic engineering have never existed in billions of years of evolution. They consist of genetic material originating from bacteria, virus, and other genetic parasites that cause disease and spread drug antibiotic resistant genes. They are designed to cross all species barriers and to invade genomes. The spread of such genes and gene constructs have the potential to make infectious diseases untreatable and to create new viruses and bacteria that cause disease. Horizontal gene transfer may spread transgenes to the entire biosphere. Horizontal gene transfer is the transfer of genetic material between cells or genomes belonging to the unrelated species by process other than usual reproduction. In the usual process of reproduction, genes are transferred vertically from parent to offspring, and such a process can occur only within a species or between closely related species. Bacteria have been known to exchange genes across species barriers in nature. There are three ways in which this is accomplished. In conjunction, genetic material is passed between cells in contact. In transduction, genetic material is carried from one cell to another by infectious virus, and in transformation, the genetic material is taken up directly by the cells from its environment. For horizontal gene transfer to be successful, the foreign genetic material must become integrated into the cell genome or become stably maintained in the recipient cell in some other form. In most cases, foreign genetic material that enters a cell by accident especially if it is from another species, will be broken down before it can incorporate into the genome. Under certain ecological conditions which are still poorly understood, foreign genetic material escapes being broken down and become incorporated in the genome. For example, heat shock and pollutants such as heavy metals can favor horizontal gene transfers, and the presence of antibiotics can increase the frequency of horizontal gene transfer 10 to 10,000 fold. While horizontal gene transfer is well known among bacteria, it is only within the past 10 years that its occurrence has become recognized among higher plants and animals. The scope for horizontal gene transfer is essentially the entire biosphere, with bacteria and viruses serving both as intermediaries from gene trafficking and as reservoirs for gene multiplication and recombination, the process of making new combinations of genetic material. There are many potential routes for horizontal gene transfer to plants and animals. Transduction is expected to be a main route, as there are many viruses which infect plants and animals. Recent research in gene therapy indicates that transformation is potentially very important for cells of mammals, including human beings. A great variety of naked genetic material are rapidly taken up by all kinds of cells simply as the result of being applied in solution to the eye, or rubbed into the skin, injected, inhaled, or swallowed. In many cases, the foreign gene constructs become incorporated into the genome. Direct transformation may not be as important for plant cells, which generally have a protective cell wall, but soil bacteria belonging to the genus Agrobacterium are able to transfer the T tumor segment of its tumor, including Ti plasmid, see below, into the plant cell in a process resembling conjunction. This tDNA is widely exploited as a gene transfer vehicle into plant genetic engineering. Foreign genetic material can also be introduced into plant and animal cells by insects and arthropods with sharp mouth parts. In addition, bacterial pathogens which enter plant and animal cell may take up foreign genetic material and carry it into the cells, thus serving vectors for horizontal gene transfer. There are almost no barriers preventing the entry of foreign genetic material into the cell of probably any species on Earth. The most important barriers of horizontal gene transfers operate after the foreign genetic material has entered the cell. Most foreign genetic materials, such as those presented in ordinary food, will be broken down to generate energy and building blocks for growth and repair. There are many enzymes which break down foreign genetic material, and in the event that foreign genetic material is incorporated into the genome, chemical modification can still put it out of action and eliminate it. However, viruses and other genetic parasites such as plasmids and transpoons have special genetic signals and probably overall structure to escape being broken down. A virus consists of genetic material generally wrapped in a protein coat. It sheds its overcoat on entering a cell and can either hijack the cell to make many more copies of itself, or it can jump directly into the cell's genome. Plasmids are pieces of free, usually circular genetic material that can be identified, maintained in the cell separately from the cell genome. Transpoons, or jumping genes, are blocks of genetic material which have the ability to jump in and out of genomes, with or without multiplying themselves in the process. They can also land in plasmids and be propagated there. Genes hitchhiking in gene parasites Virus plasmids and transpoons therefore have a great probability of being successfully transferred into cells and genomes, gene parasites, and vectors for horizontal gene transfer. Natural genetic parasites are limited by species barriers. So, for example, pig viruses will infect pigs, but not human beings, and cauliflower viruses will not attack tomatoes. It is the protein coat of the virus that determines host specificity, which is why naked viral genomes, the genetic materials stripped of the coat, have generally been found to have a wide host of range that the intact virus. Similarly, the signals for propagating different plasmids and transpoons are usually specific to a limited range of host species, although there are exceptions. As more and more genomes have been sequenced, it is becoming apparent that gene trafficking or horizontal gene transfers has played an important role in the evolution of species. However, it is also clear that horizontal gene trafficking is regulated by internal constraints in the organisms in response to ecological conditions. Gene engineering is unregulated horizontal gene transfer. Gene engineering is a collection of laboratory techniques used to isolate and combine the genetic material of any species, and then to multiply the constructs in convenient cultures of bacteria and viruses in the laboratory. Most of all, the techniques allow genetic material to be transferred between species that would never interbreed in nature. That is how many genes can be transferred into pig, sheep, fish, and bacteria. Spider silk genes end up in goats. Completely new exotic genes are also being introduced into food and other crops. In order to overcome natural species barriers limiting gene transfer and maintenance, gene engineers have made a huge variety of artificial vectors, carriers of genes by combining parts of the most infectious and natural vectors, viruses, plasmids, and transpoons, from different sources. These artificial vectors generally have their disease-causing functions removed or disabled, but are designed to cross wide species barriers, so the same vector may now transfer, say, human genes spliced into the vector to genomes of all other mammals or of plants. Artificial vectors greatly enhanced horizontal gene transfer. Artificial vectors enhanced horizontal gene transfers. They are derived from natural genetic parasites that mediate horizontal gene transfer most effectively. Their highly chromatic nature means that they have sequence homologies, similarities to DNA from variable pathogens, plasmids, and transpoons of multiple species across kingdoms. This will facilitate widespread horizontal gene transfer and recombination. They routinely contain antibiotic-resistant marker genes which enhance their successful horizontal transfer in the presence of antibiotics, either intentionally applied or present as xenobiotic in the environment. Antibiotics are known to TQ enhance horizontal gene transfer between 10 to 10,000-fold. They often have origins of replication and transfer sequences signals that facilitate horizontal gene transfer and maintenance in cells to which they are transferred. Chimeric vectors are well known to be structurally unstable, i.e. they have tendency to break and join up incorrectly or with other DNA, and this will increase the propensity for horizontal gene transfer and recombination. They are designed to invade genomes, to overcome mechanisms that break down or disable foreign DNA, and hence will increase the probability of horizontal transfer. Although different classes of vectors are distinguishable on the basis of the mainframe genetic material, practically every one of them is chimeric, being composed of genetic material originating from the genetic parasites of many different species of bacteria, animals, and plants. Important chimeric shuttle vectors enable genes to be multiplied in the bacterium E. coli and transferred in species in every other kingdom of plants and animals, simply by creating such a vast variety of promiscuous gene transfer vector genetic engineering biotechnology has effectively opened up highways for horizontal gene transfer and recombination where previously the process was tightly regulated. With restricted access through narrow, tortuous footpaths, these gene transfer highways connect species in every domain and kingdom with the microbial populations via the universal mixed vessel used in genetic engineering E. coli. What makes it worse is that there is currently still no legislation in any country to prevent the escape and release of most artificial vectors and other artificial constructs into the environment. What are the hazards of gene transfer? Most artificial vectors are either derived from viruses or have variable genes in them and are designed to cross species barriers and invade genomes. They have the potential to recombine with the genetic material of other viruses to generate new infectious viruses that cross species barriers. Such viruses have been appearing at alarming frequencies. The antibiotic-resistant gene carried by artificial vectors, resurgence of drug and antibiotic infection diseases with the past 25 years. There is already overwhelming evidence that horizontal gene transfer and recombination have been responsible for creating new viral and bacterial pathogens and for spreading drug and antibiotic resistance among the pathogens. One way that new viral pathogens may be created is through recombination with dormant, inactive, and inactivated viral gene material that are in all genomes, plants and animals without exception. Recombination between external and resident dormant viruses have been implicated in many animal cancers. As stated earlier, the cells of all species, including our own, can take up foreign genetic material. Artificial constructs designed to invade genomes may well invade our own. These insertions may lead to inappropriate inactivation or activation of genes, insertion mutagenesis, some of which may lead to cancer, insertion carcinogenesis. Hazards of horizontal gene transfers are summarized. Potential hazards of horizontal gene transfers from genetic engineering. Generation of new cross-species viruses that cause disease. Generation of new bacteria that cause diseases. Spreading drug and antibiotic resistant genes among the viral and bacterial pathogens, making infections untreatable. Random insertion into genomes of cell resulting in harmful effects including cancer. Reactivation of dormant viruses presents in all cells and genomes which may cause diseases. Spreading new genes and gene constructs that have never existed. Multiplication of ecological impacts due to all of the above. Transgenic DNA may be more likely to transfer horizontal than non-transgenic DNA. Both the artificial vectors used in genetic engineering and the genes transferred to make transgenic organisms are predominantly from viruses and bacteria associated with disease, and these are being brought together in combinations that have never existed in billions of years of evolution. Genes are never transferred alone. They are transferred in unit constructs known as expression cassettes. Each gene has to be accompanied by a special piece of genetic material. the promoter which signals the cell to turn the genome on. In other words, the transcribe that DNA gene sequence into RNA. At the end of the gene, there has to be another signal, a terminator, to end the transcription and to mark the RNA so it can be further processed and translated into protein. The simplest expression, cassette, looks like this: promoter, gene, terminator. Typically, each bit of the construct promote, gene, and terminator is from the different source. The gene itself may also be composite of bits from different sources. Several expression cassettes are usually linked in series, stacked in final construct. At least one of the expression cassettes will be that of the antibiotic-resistant marker gene-enabled cell that have taken up the foreign construct to be selected with antibiotics. The antibiotic-resistant gene cassette will often remain in the transgenic organism. The most commonly used promoters are from viruses associated with serious diseases. The reason is that such viral promoters give continuous overexpression of genes placed under their control. The same basic construct is used in all applications of genetic engineering, whether in agriculture or in medicine. The same hazards are involved. There are reasons to believe that transgenic DNA is much more likely to spread horizontal than the organism's own DNA. Reasons to suspect that transgenic DNA may be more likely to spread horizontally than non-transgenic DNA. Artificial constructs and vectors are designed to be invasive to foreign genomes and overcome species barriers. All artificial gene constructs are structurally unstable, 20, and hence prone to recombine and transfer horizontally. The mechanisms enable foreign genes to insert into the genome, also enable them to jump out again, to reinsert in another site, or to another genome. The integration sites of most commonly used artificial vectors for transferring genes are recombination hotspots, and so have increased propensity to transfer horizontally. Viral promoters, such as that from the cauliflower mosaic virus, widely used to make transgenes overexpress, contain recombination hotspots and will therefore further enhance horizontal gene transfer. The metabolic stress on the host organism due to the continuous overexpression of transgene may also contribute to the instability of the insert. 22. The foreign gene constructs and the vectors into which they are spliced are typically mosaic of DNA sequences from numerous species and their genetic parasites. That means they will have sequence homologies with the genetic material of many species and their genetic parasites, thus facilitating wide-ranging horizontal gene transfer and recombination. Additional hazards from viral promoters. We have recently drawn attention to the additional hazards associated with promoter of cauliflower mosaic virus, CAMV, most widely used in agriculture. 23. It is particularly all transgenic plants already commercialized or undergoing field trials, as well as high proportion of transgenic plants under development, including the much acclaimed golden rice. 24. CAMV is closely related to human hepatitis B virus and less so to the retrovirus such as the AIDS virus. 25. Although the intact virus itself is infectious only for cruciferous plants in promoter in promiscuous in function and is active in all higher plants in algae, yeast, and E. Coil 26. As well as frog and human cell systems. 27. Like all promoters of viruses and cell genes, it has a modular structure with parts of common to and interchangeable with promoters of plants and animal viruses. It is a recombination hotspot flanked by multiple motifs involved in recombination similar to other recombination hotspots, including the broaders of Agrobacterium. tDNA, vector most frequently used in making transgenic plants. The suspect mechanism of recombination requires little or no DNA. Generate new virus 28. In some cases, the recombinant viruses are more infectious than the original. Proviral sequences, generally inactive copies of viral genomes, are present in all plants and animal genomes, and all viral promoters are module and have at least one module, the TATA box, in common, if not more. It is not inconceivable that the CAMV35S promoter in transgenic construction can reactivate dormant viruses or generate new viruses by recombination. The CAMV35S promoter has been joined artificially to copy of a wide range of viral genomes and infectious virus produced in the laboratory. There is also evidence that proviral sequence in the genome can be reactivated. The considerations are especially relevant in light of recent findings that certain transgenic potatoes containing CAMV35 promoter transformed with Agrobacterium tDNA may be unsafe for young rats, and that's a significant part of the effects may be due to the construct of genetic transformation or both. 31. The authors also report an increase in lymphocytes in the intestinal wall, which is non-specific sign of viral infection. Evidence of horizontal transfer and transgenic DNA. It is often argued that transgenic DNA, once incorporated into the transgenic organism, will be just as stable as the organism's own DNA, but there is both direct and indirect evidence against this suspicion. Transgenic DNA is more likely to spread and has been found to spread by horizontal gene transfer. Transgenic lines are notoriously unstable and often do not breed true. There is particularly of molecular data documenting the structural stability of transgenic DNA, both in terms of its site of insertion in the genome and its arrangement of genes in successive generations. Instead, transgenes may be silenced in subsequent generations or lost altogether. Herbicide tolerance gene introduced into Arabidopsis by means of vector was found to be up to 30 times more likely to escape and spread than the same gene obtained by mutagenesis. One way this may happen is by second horizontal gene transfer via insects visiting the plants from pollen and nectar. The reporting finding that pollen can transfer transgenic DNA to bacteria in the gut of a bee larva is relevant here. Second, horizontal transfer of transgenes and antibiotic-resistant marker genes from genetically engineered crop plants into soil bacteria and fungi have been documented in the laboratory. Transfer to fungi was achieved simply by company cultivation 37, while transfer to bacteria has been achieved by both re-isolated transgenic DNA or total transgenic plant DNA. Successful transfer of a kanamycin-resistant marker gene to the soil bacterium Acinetobacter were obtained using total DNA extracted from homogenized plant leaf from a range of transgenic plants: Solanum tuberosum, potato; Nicotiana tabacum, tobacco; Beta vulgaris, sugar beet; Bacillus neplis oilseed, Lycopodium esculentum tomato. It is estimated that about 2,500 copies of Kanamacean resistant genes from the same number of plant cells is sufficient to successfully transform one bacterium, despite the fact there is 6 million-fold excess of plant DNA present. A single plant with, say, 2.5 trillion cells would be sufficient to transform 1 billion bacteria. Despite the misleading title in one of the publications, a higher gene transfer frequency of 5.8 by 10 to the second per recipient bacterium was demonstrated under optimum conditions, but the authors then proceeded to calculate an extremely low gene transfer frequency of 2.0 by 10 to the 17 under extrapolated natural conditions, assuming that different factors acted independently. The natural conditions, however, are largely unknown and unpredictable, and even by authors' own admissions, synergistic effects cannot be ruled out. Free transgenic DNA is bound to be readily available in rhizosphere around the plant roots, which is also an environmental hotspot for gene transfer. Other workers have found evidence of horizontal transfer of Kanamacean resistance from transgenic DNA to Acinetobacter, and positive results were obtained using just 100-yol plant leaf homogenate. Defenders of the biotech industry still insist that just because horizontal gene transfer occurs in the laboratory does not mean it can occur in nature. However, there is already evidence suggesting it can occur in nature. First of all, genetic material released from dead and live cells is now found to persist in all environments and not rapidly broken down as previously supposed. It sticks to clay, sand, and humic acid particles and retains the ability to, in fact, transform range of microorganisms in the soil. The transformation of bacterium in the soil by DNA adsorbed to clay, sand, and humic acid has been confirmed in microcosm experiments. Researchers in Germany began a series of experiments in 1993 to monitor field releases of transgenic rhizomania-resistant sugar beet, Beta vulgaris, contained the marker gene for Kanamacean resistance for persistence of transgenic DNA and of horizontal gene transfer of transgenic DNA into soil bacteria. It is the first such experiment to be carried out after tens of thousands of field releases and tens of millions of hectares have been planted with transgenic crops. It will be useful to review the findings in detail.